Today my topic is Paul want me to actually talk about the current research studies in the axial SPA. And I thought that we will probably firstly just briefly talk about what we sort of know about the driver of the disease, and then later we will actually talk about what are the sort of approaches that we have been using trying to understand the disease, and then finally I will give you a showcase of one of the projects that we have been carrying on for this goal. So as Paul mentioned earlier, that it's really strongly affected by genetic risk, and HLA-B27 is really the top one on the list. And then apart from that, we know that it's really driven by inflammation in white blood cells. And then as Claudia mentioned earlier, several pro-inflammatory proteins such as TNF-alpha, interleukin-17, and Janus kinase are available biological targets. And then I think all of us know that not everyone responds well to those treatments, and actually sometimes you might lose the response to the drug that used to work for you, and very importantly, the long-term remission or cure hasn't been achieved. So that's why we want to solve this problem through our work. And then so there are several, many approaches that we have been undertaking to understand the disease and to carry out the research. And it can be broadly divided to 4 parts. The first part is to use the blood or joint tissue to understand the condition. And then we are using various of the cutting-edge technologies for this purpose. And then the showcase project that I'm going to show you later use one of the most advanced technology to understand the feature of the cells at the single cell level. And then the second approach is in vitro models. And then we try really hard to generate in vitro cellular model that can sort of be as close as possible to what's going on in the human body, and then especially to the pathology that's going on in a swollen knee or swollen joint. And then this part is really difficult, but we have been trying to do so. And then the third part is something that is almost crucial for the development of the new drug, that's in vivo mirroring models. A general consensus is that in the field now is that this model is not very good to understand the disease because it's artificial, it's introduced in a certain manner, and then those drivers of those models might not be relevant to disease. However, because we know what drove those diseases, and if we find a biology using the first approach, using the blood and joint from the patient and from the inflamed tissue, then we know what's driving it, and then we can use the relevant model to test if certain drug can actually suppress it in an in vivo setting. So we mostly use it for that purpose. And then lastly, and very importantly, all this a new drug need to be tested in clinical trial before we know that it's actually something that's really going to be useful. I'm going to give you an example of the project, and this project is actually really based on the in vitro cellular models using the cells from the blood. And then so this is actually what we do with the blood that you donated. And then we firstly, on the left, we use a reagent that sort of have higher density than the blood. So as a result, it's actually sitting at the bottom. And then blood is at the top. And then after the centrifugation, based on because different components of the blood have different density. And then based on the density, we're able to separate the white blood cells that are highlighted in blue. And that's actually where all those sort of bad cells that are producing the IL-17 by T cells, as Claudia said earlier, and also TNF by monocyte or macrophage, it's all there. And then so we firstly separate those white blood cells, and then after that, for this particular project, we use a technology called single-cell RNA sequencing, and then this is relatively new. But what the information they tell us is that it's sort of a group the cells based on its feature, and then the cells that are different from each other, they will be separated differently. And then here, it's actually very, very simple. Essentially, the cells in different each dot is a cell, and each population in different color, they are actually population that's different from other populations, and then they will have different functions. And then the cells that we found to be very inflammatory, pro-inflammatory, is this sort of a green or yellow-green color, and then these cells we call them trained monocyte. And the reason we call them trained monocytes is because they share some of the feature with a group of the monocyte that has received stimulation, and then they are more pro-inflammatory compared to the rest of the monocytes. So it's called trained monocyte. But the name is not important. Why it's important is that if it's relevant to axial SPA or not. And then so here we use a data set to look at the percentage of this population of cells, comparing the healthy control in sort of red or pink versus the AS in green. And so you can see that this population of the cells are actually significantly increased in the individuals carrying this AS condition. And then, but we don't know whether this is causal. Could this be actually a consequence of disease, or could this be actually an increase the drive for the disease? And this is really important. If it's consequence, then targeting or studying those population might not really help. So to answer this question, we test one of the most important genetic factor and a protein that you are very familiar with, HOA-B27. So we carry out this using a big cohort of healthy control. This cohort has more than 1,000 people, but they are all healthy. But within this cohort, there are about 100 of the healthy donors are carrying HOA-B27. So when we look at this donor, this group, then we can actually see the abundance of this so-called trained monocyte is also increased in the B27 positive healthy donors. And then the best control is probably the others, because the reason that we separate the B51 and B7, because both of them actually also is associated with either increased or reduced risk of the developing the condition. So it's sort of confounding factor, we need to take it out. And then, but again, this is just an association. And to test if B27 is really driving the expansion or increase of this subset of the monocyte, we carry out an experiment. It's actually genetically suppress the expression of this gene HOA-B27. Now what we found is that all the signatures coming from that trained monocyte are reduced when we suppress the expression of this HOA-B27 gene. So this is a very important evidence for us to start to believe that B27 could actually be driving the expansion of this trained monocyte. And then if that's the case, then understanding this trained monocyte and then targeting those trained monocytes might actually help us to control the disease a bit better. So what to do next? What we will do is that we'll try to isolate those trained monocytes out, and then try to study and see that whether they are really like produce high amount of the bad cytokines to the stimulation, and then or if we put them into a sort of a in vitro cellular model, are they going to do actually worse than the other cells? And then second point, which is really, really important, is that can we find a way to inhibit or reprogram or even deplete those cells? And one of the possible ways, actually, if we can establish the B27's driving role in the expansion of those cells, then maybe HOA-B27 is the target. and then maybe we can just target HLA-B27. And then lastly, I want to actually highlight it that the groups in Oxford have been really working very, very close together. And then this project was carried out in close collaboration with Paul's lab. And then so Feng and Hui, they have actually worked together in this project. And then we have been working also with the other scientists in Oxford and also across the world, trying to understand the disease better and hopefully it can improve the treatment and bring a cure. Thank you.